Dear students, so in the last class we have seen something about the organelles of eukaryotic cells, and we have seen something about the endomembrane systems. Okay, and today we are going to start with the some other organelles that are present outside the endomembrane system, and we are going to start with mitochondria. So what is mitochondria and what is the role or the functions played by the particular organelle in the eukaryotic cells? Okay, so the mitochondria already we know they are the powerhouses of the cells. So they are the major organelles. They are participating in the aerobic respiration or the production of the ATP or the energy carrier molecules of the cell. Okay, so the number of mitochondria per cell varied based on a type of cells. Normally their numbers varied from 1 to some thousands of mitochondria per cell. Okay, so their shape also varied based on a cell. Their shape may be sausage shaped or cylindrical shaped. Their size approximately in diameter they will be around 0.2 to 1 micrometer in diameter. Okay, in diameter they will be around maximum of 1 micrometer in diameter, average 0.5 micrometer in diameter. The lengthwise, okay, it will be around 1 to 4.1 micrometer in length. Okay. So now we will see something about the structural details of the particular mitochondria. So the mitochondria already we know the organelles are membrane bound. Okay, so you have to remember this point. All the organelles are membrane bound. Okay, so the mitochondria is being covered with the two layered or two membranes. Okay, so the outer membrane and inner membrane. So the outer membrane limits the size of the particular mitochondria. The inner membrane, it is present inside the particular mitochondria. Okay, so it has so many invaginations towards the matrix. Matrix is a semi-liquid substance present inside the mitochondria. Okay, so there are so many invaginations present inside the inner membrane towards the matrix. So what is the purpose of the particular invagination? So that particular invagination enhances or increases the surface area. Okay, so this invagination forms a structure called the finger-like structures or cilia-like structures called crista or cristae. Okay, cristae is a plural and crista is a singular. Crista or cristae. Okay, so these invaginations increases the surface area of the particular mitochondria. So the matrix also contains so many other molecules such as DNA, RNA and ribosomes. They all involved in the protein synthesis. Okay, so you must remember one thing. There are some hypotheses. Okay, so the mitochondria are actually the descendants of bacteria. To support the particular hypothesis, there are some points you have to remember. The first thing, the mitochondria possess covalently closed circular DNA. Okay, we all know the eukaryotic cells possess the linear kind of DNA. They won't have the circular DNA, but mitochondria, they have their own DNA. The form of DNA is covalently closed circular DNA. And the one more point you have to remember, normally we know the ribosomes in eukaryotic cells, they are 80S ribosomes, but within the mitochondria, ribosomes are 70S ribosomes. So the point you have to remember over here, the mitochondria, they have their own DNA, they have their own ribosomes, they are producing their own enzymes required for the respiration or the powerhouse purpose. Okay, so I told you there are two membranes, outer membrane and inner membrane. There is a space in between the outer membrane and inner membrane. That space is called intermembrane space. Okay, so both the membrane, they have their own proteins and enzymes, and in between the membranes, that is the intermembrane space also contains some enzymes. All the enzymes are participated in the respiration or the ATP production. One more point you have to remember, the mitochondria is dividing by means of the binary fission. So normally only the prokaryotic cells will perform the particular binary fission or binary division, and the mitochondria, as I told you, as it is a descendant of a bacteria, They are also dividing by means of the binary fission. Okay. So the purpose or role of mitochondria, as I told you, it is involved in aerobic respiration. You have to remember this. Aerobic respiration. They are going to use the oxygen as a electron acceptor to produce the final electron, the energy carrier molecule, ATP molecules. Okay. So the points you have to remember for the mitochondria are the powerhouses of the cell. They are going to produce the energies in the form of ATP molecules. Their shape may be varied in the sausage or cylindrical shape. Their size, the diameter may be 0.2 to 1 micrometer. Their length may be 1 to 4 micrometer. Okay. So there are two membranes present in the mitochondria, outer membrane and inner membrane. Both the membrane possess so many enzymes. They are involved in the respiration. The space in between the membranes are called intermembrane space, and within an inner membrane there is a matrix. That matrix possesses so many molecules. The major molecules present in the matrix are 70S ribosomes, covalently closed circular DNA, and so many RNA molecules. They all involved in the synthesis of proteins required for the roles played by the particular mitochondria. There are structures called cristae. They are nothing but the invaginations of the inner membrane. They are increasing the surface area so that they are increasing the production of the ATP molecules. And next structure we are going to see now, the plastids. Now we are going to see something about the next organelle, the plastids. Okay. So the plastids, you have to remember the major point. The plastids are only present in plant cells and euglenoids. Euglenoids, all we know, they are the primitive, the protozoan groups. So they are present only in the plant cells and euglenoids. So they normally possess some pigments. Because of the pigments, they are providing some colors to the parts of the particular plants they are having. Okay. So next, there are so many categories of the plastids present, and we are going to see one by one. We are going to start with the chloroplast. Okay. So the chloroplast, all we know, they are possessing the pigments called carotenoids and chlorophylls. Okay, carotenoids and chlorophylls. What is the purpose of the chloroplast? Chloroplast, they are involved in the photosynthesis. Okay, they are trapping the light energy from the sunlight, and they are converting the particular light energy into the storage molecule called the carbohydrate. Okay, so the chloroplast possessing the chlorophylls and carotenoids, these pigments, they have the power of trapping the light energy. They are involved in the protein synthesis, the photosynthesis, and they are forming the molecules or the storage molecules, the carbohydrates. So the next group of plastids we are going to see, the chromoplast. So what are chromoplasts? Chromoplasts also contain some pigments, carotenoids, okay, fat soluble carotenoids. The purpose of carotenoids over here, they are giving the color for the particular plant based on carotenoids. There are so many different types of carotenoids available, carotene, xanthophylls, okay. So based on carotenoids present in the particular plant, they are providing some colors to the part of the plant, yellow color, orange color, red color, okay. So all the color, whatever may be the color you are seeing in the particular plant, that particular color is being provided by the pigments of the particular plastids. If it is in a green color, it is being provided by the chloroplast, that pigments are the chlorophylls and carotenoids. If it is in a red color, yellow color, orange color, that is being provided by the chromoplast pigments, okay. So the third category, the leucoplast. So this is a colorless pigments, okay. So these pigments are colorless plastids or colorless pigments. So over here there are three major categories or divisions available: amylopectins, elipeptins, and alleroplast. So amylopectins over here, they are gonna store the carbohydrate molecule. I told you they won't provide the color for the particular part of the plant. They are going to provide the storage purpose of the particular plant. So over here the amylopectin, they are going to store the carbohydrate molecule as it is doing in the potato. And elaiplast, they are going to store the oils and the fats and aeroplast, they are gonna store proteins for the particular plant. And now we are going to see the structural details of a particular type, chloroplast. So as we all know, the chloroplasts are involved in the photosynthesis, as the mitochondria involved in the respiration. So over here, without chloroplast, there will be no photosynthesis, or there will be no synthesis of carbohydrate in plants. Okay. So as I told you, as in the other organelles, this is also made up of the membrane bound structure. So there are two membranes, outer membrane and inner membrane. There are two membranes covered the particular structure called the chloroplast. So within the inner membrane there is a fluid or matrix like structure called stroma. So within a stroma there are structures called thylakoid. Okay. So what is thylakoid? Thylakoid is there are structures. There are some disc shaped structures. So this structure is called granum. So this granums are piled up one above each other and they will form the structure called thylakoid. So these thylakoid are interconnected with each other by means of the lamellae called stromal lamellae. Within the thylakoid there is a space present that is called the thylakoid lumen. So in the structure you have to remember these points only. There are membranes, outer membrane and inner membrane. Within the membrane there is a matrix called the stroma. Okay. And within the stroma there are structures called granum. They are coin shaped structures, and these granum are arranged or piled one above each other and they will form the structure called the thylakoid, and thylakoid connected with each other by means of the tubular structure. Okay, tubular structure, and these structures are called stromal lamellae. Okay, so apart from these thylakoid, and there are so many macromolecules also present in the particular stroma. As I told you in the mitochondria, it is also, the chloroplasts also descendants of the bacteria, and they are also possessing their own covalently closed circular DNA. They are also possessing their own RNA molecules, and they are also possessing their own enzymes. And of course, the unique ribosomes, 70S ribosomes. Just remember, in case of eukaryotic cells, all the ribosomes are 80S ribosomes, but only in mitochondria and chloroplast, the ribosomes are 70S ribosomes because they are descendants of bacteria. So they are possessing the characters of bacteria, 70S ribosomes, circular DNA, their own RNA, their own enzymes. So they are producing their own proteins, the proteins required for the photosynthesis. Okay. So the points you have to remember in the plastids, there are so many different types of plastids available. They are available only in the plant cells and euglenoids. So they are normally possessing the pigments, they are providing the color for the particular part of the plant. So the categories are the chloroplast, chromoplast and leucoplast. Among the three, the leucoplast are colorless plastids, and the remaining two, they are possessing the colored pigments. The chloroplast normally present in the photosynthetic areas of the plants. So what is the normal photosynthetic area of plant? Already we know, the mesophils of the growing leaves. Okay. So in a nucleus, normally the green areas of the plant, they are possessing the particular chloroplast plastids. They are possessing the pigments carotenoids and the chlorophylls. They are trapping the light energy from the sunlight to produce the carbohydrate molecule by means of the photosynthesis. Chromoplast, they are having the colored pigments such as carotene, xanthophylls. They are giving color for the part of the plant, yellow, orange and red. And the colorless plastids are leucoplast. There are three categories, major categories: amylopectins, alloyplast and alluroplast. They are storing the carbohydrate molecules, oils, fats and proteins. And we have seen the structural details of the chloroplast, two membrane structures and stroma, internal structures, thylakoid connected with the tubular structures are stromal lamellae. Apart from these structures, there are molecules as in the mitochondria and bacteria, covalently closed circular DNA, their own enzymes and 70S ribosomes. Okay, and now we are going to see something about the next major organelles of eukaryotic cells, the nucleus. And now we are going to see one of the major organelles of the eukaryotic cells, the nucleus. So as I told you in the previous classes, the nucleus is the organelle which is going to be a major point of the differences between the prokaryotic and the eukaryotic cells. So the nucleus, it is being covered by the membrane. So as in the other organelles, nucleus is also covered by the membrane bound structures. So there are two membranes, inner membrane and outer membrane, and we are going to see the structural details now. Okay. So before that, you should know something about the history of nucleus. As I told you in the previous classes, nucleus is being discovered by Robert Brown in the year 1831, and the chromatin, the name is being given by the scientist called Fleming, and who has discovered the staining procedure for the DNA molecules or the cells internal structures called the nucleus, and he has discovered the structures present in the nucleus and he has named it as a chromatin. Okay, and what are chromatins? The chromatins are the elongated fiber-like structures present all over the particular nucleus, and you can see the chromatin structures only in the stage called interphase. So what is interphase? Already we know during the cell division cycle, so the non-dividing stage, that is the cells are in the resting phase. So during the non-dividing stage or the resting phase, you can observe the chromatin like fibers in the particular eukaryotic cells nucleus. Okay, so chromosomes. So what are chromosomes? During the other phases of cell division, during the active cell division stages, you can observe the structures called chromosomes. Both the chromatin and the chromosomes are made up of DNA along with the proteins called histones. So what are histones? Histones are nothing but the packing proteins. So histones are only present in the eukaryotic cells, they are absent from prokaryotic cells. So histones are packing proteins, they are going to pack the entire DNA molecules into a structures called chromatin or chromosomes along with some other non-histone proteins. And now we are going to see the basic structure of any nucleus. As I told you, it is made up of two membranes, outer membrane and inner membrane. Okay, so the outer membrane, as you can see it over here, outer membrane is actually a continuation from the endoplasmic reticulum. Okay, so actually the outer membrane is being connected with or the continuation with the endoplasmic reticulum that is present or immediately present outside the particular nucleus structure, and the inner membrane, it is the limiting structure of the particular internal structure of nucleus, and within the inner membrane there is a matrix. Matrix, as I told you, is a semi-liquid structure. The matrix is present, nucleomatrix. So within a matrix, the liquid portion is called the nucleoplasm. So within a nucleoplasm, there are so many structures or molecules present all over the nucleoplasm, and the major structure you can easily observe under the microscope is nucleolus. Okay, nucleoli is the plural. So the nucleolus involved in the rRNA synthesis, that is synthesis of the ribosomal subunits. Synthesis of ribosomal subunits. We are going to see the details of ribosomes of eukaryotes later. The point you have to remember over here, within a nucleoplasm there is a distinct structure present over here. That structure is called nucleolus. That nucleolus involved in the synthesis of ribosomal subunits. What are ribosomal subunits? They are made up of rRNA plus number of proteins. So they are involved in the synthesis of smaller ribosomes and a larger ribosomal subunit. So apart from nucleolus, as I told you, the structures, the chromatin you can easily observe under the microscope. So the chromatin you can see only in the stage called interphase. In other stages, in actively dividing cells, you can see only the structures called the chromosomes, the constricted or packed structure of DNA along with the histone molecules or histone proteins. So apart from DNA, apart from chromatin, whenever you are seeing the term chromatin or chromosome, and you have to remember the term DNA. DNA is packed as a chromatin or chromosome over here. So apart from DNA molecules, there are structures called RNA molecules. So wherever the DNA is present, the RNA will also present in the same place, because the RNA is being transcribed from or synthesized from the particular DNA molecule. Okay, So the RNA molecules also present and so many different types of the proteins also present all over the particular nucleoplasm. Okay. So and one more space you have to remember, the space in between the inner membrane and outer membrane, that space is called the periplasmic space. That periplasmic space also contains so many different types of enzymes for the nucleus function. Apart from periplasmic space, the membrane also possesses some pores, some holes. Those pores are called nuclear pores. So what may be the purpose of the particular nuclear pore? Through the particular pores only, the synthesized RNA molecules move out of the particular nucleus and the proteins move into the particular nucleus. The movement of RNA and proteins in and out of the nucleus, that is happened are through the particular nuclear pores present in the nuclear membranes. Okay. So what are the structures you have to remember in case of the nucleus? There are two membranes, outer membrane and inner membrane. Between the two membranes there is a space called periplasmic space. The outer membrane is being continuous with the endoplasmic reticulum. So apart from this, there is a matrix called nuclear matrix. Within the matrix, liquid portion called nucleoplasm. Within the nucleoplasm, nucleolus, chromatin and RNA molecules are present, and nuclear pores are involved in the exchange or transport of RNAs and proteins in and out of the particular nucleus. Okay. So I told you that chromosomes you can see it only in the cell dividing stage. Okay. So in the chromosomes you can see the clear structure called centromeres. Okay. So it will be like this. So this central structure is called centrosomes or centromeres, and the centromeres contains the disc shaped structures called kinetochores. Okay, so normally they are involved in the cell division, the equal division of DNA molecules in between the daughter cells during the cell division. Okay, so based on a position of or the location of or the site of centromere, we can divide the chromosomes into four types. There are four types of based on the position of centromere. Okay, so metacentric, submetacentric, acrocentric and telocentric. So what is metacentric? Over here, the position of centromere is almost equal between the chromatids. Okay, so you can see the equal length of chromatids present in the both the sides of the particular centromere. Submetacentric, you can see it over here, unequal distribution of the chromatids. Okay, one side of the centromere is a shorter chromatids and other side they will have the larger chromatids. Acrocentric, over here you can see the differences, even much larger differences. Okay, over here the larger chromatids on one side and the very smaller chromatids on the other side. Telocentric, almost one side is absent and the other side is larger than this side. Okay, so over here in case of telocentric, the centromeres are almost towards the terminal. Okay, so based on the position of centromere, we are dividing the entire chromosomes into four types: metacentric, submetacentric, acrocentric and telocentric. So apart from this, you have to observe another structure in the chromosomes called satellites. Okay, in some chromosomes there are some secondary constrictions like this. They are secondary constrictions. Okay, and they will form some fragment like appearance of structures in the terminal region of chromatids, and these fragment like structures called satellites. Okay, and these are the things you have to remember whenever you are thinking about a nucleus. So nucleus is matter, memory of the cells. So they are possessing all the information for the cells. They are the major organelle of any cell, any eukaryotic cells. Okay, so they are being discovered by Robert Brown and they are being stained by Fleming. And the structures you have to remember in the nucleus: outer membrane, inner membrane, periplasmic space and within that nucleolus, nucleoplasm, and within the nucleoplasm chromatin, RNA molecules and some other protein molecules you have to remember, and the major structures present in the membrane called nuclear pores, and the pores involved in the exchange of or the transport of RNA and proteins in and out of the particular nucleus. In case of chromosomes, you have to observe the structure called centromere and satellites. Centromere which connects the chromatids, satellites is a secondary constructions. Based on a position of centromere, there are 4 types of chromosomes we can observe over here. Okay, so in the next class we are going to see the remaining organelles and the remaining structures of the eukaryotic cells. Then we are going to see the question pattern we can expect from the particular eukaryotic cell structures and the prokaryotic cell structure and its functions.